Good evening, my friends. Welcome back to our channel once again. And today we're going to discuss cell components, the different components wey dey inside cell, and wetin be the usefulness of all these different components, wetin they help us to do. Now, for some of the former videos wey we don do, you'll see we don't talk about the classification of cells. We say one man dey wey im name be Carolus Linnaeus, na Swedish man, and him be botanist, him dey study plants. Na him divide all the organisms wey dey for this world into two major kingdoms. Him divide them into plants and animals. So today we wan look at the different cells, components of those plant and animal cells. We will come look at inside those cells, look at wetin dey inside the cells, wetin dey make the cells function, and wetin dey make the cells, carry out all their life processes. This one dey very important. As a scientist, you need to understand wetin dey for inside cell, whether na plant cell or na animal cell, and the little differences wey bin dey inside them. Now, the first thing wey we go start with na the nucleus. Wetin the nucleus be? The nucleus na structure wey dey circular. E dey like circle, like bag, and e dey for inside both plant cell or animal cell. And wetin the nucleus dey do? The nucleus dey carry all the genetic information, all the genetic materials wey the cell get, e dey inside the nucleus of the cell. So the nucleus dey very important. Both plant cell and animal cells dey get nucleus. Now, after nucleus, the second thing wey we go talk about na chromosomes. Wetin chromosome be? Chromosome na structure wey resemble thread. We know this thread wey tailors dey use sew cloth or different materials. Now, chromosome, e be like those thread, but no be for sewing cloth or anything. E dey there to hold DNA in place. Now, the question will come say wetin be DNA? Many times students dey confused. Things wey dey confuse students na DNA, chromosome, chromatid. All these things, students dey always wonder say shey na the same thing. Wetin be DNA? Wetin be DNA? Wetin be chromosome? Wetin be gene? Wetin be chromatid? Now, we will explain everything for you one by one so you go understand. The first thing wey I go start with na the DNA. Wetin be DNA? Now, for inside any living cell, whether na plant cell or na animal cell, e get one giant molecule, na giant structure like that, and they been weave am like step. Na wetin oyibo man dey call double helix. They weave am like that, like where they weave woman hair. Na so the structure be. And that giant molecule, e dey carry all the information, all the genetic information. And wetin be genetic information? The way a person go look, in height, in weight, the color of him eye, the color of him skin, all those things, science don code am, they don write am for inside the DNA. So the DNA dey carry all that information, and DNA na that giant molecule wey dey carry that information. And dey important to know say the DNA na wetin we dey call polymer molecule. And the meaning of polymer molecule na say na giant molecule wey dey made up of different smaller molecules. Those smaller molecules go come together, come make that giant molecule. Now, the smaller molecule na wetin we dey call nucleotides. And those nucleotides, if you join them together, they be about 4. When we join them together, they make that giant molecule. So many nucleotides go dey like that wey dey make that giant molecule. So the nucleotide, na then they make that giant molecule wey we dey call DNA. Now, wetin come be genes? Genes na say for that giant molecule, different portions, small portions wey dey for the body of that molecule, they dey code, they dey represent different characteristics. So my eye color now, the color of my hair, my height, my skin color, all these things, they don write am down for inside the DNA for small portions of that giant molecule. Small portions for the molecule go dey carry that information. Say my hair go black, my eye go brown. All those things, they don code am for that place. And those small portions of the giant molecule wey dey carry that information na wetin we dey refer to as gene. So gene dey, DNA dey. The next thing na chromosome. So wetin chromosome come be? Like I talked before, I talk say chromosome na thread-like structure. And that thread-like structure, we dey use am, or for inside cell, e dey hold the DNA molecule in place. So e go wrap around the DNA molecule, hold the DNA molecule very well. And e no dey do that one alone. E dey do am together with something wey we dey call histones. And wetin histones be? Histones na proteins, different kind of proteins wey dey inside the nucleus. They dey together with the chromosomes, and they go hold the DNA tight, wrap themselves around the DNA. So na wetin chromosome be. So you get genes, you get DNA, you get chromosome. The fourth thing wey dey confuse students na chromatid. Wetin be chromatid? Now chromatid na say if the cell want to divide, if you reach the time wey the cell don old enough and the cell wan divide, you know different types of cell division dey. We go still discuss all those things as we dey go. Now one of the types of cell division na wetin we dey call mitosis, and for that type of cell division, one parent cell, one mama cell go divide to make two daughter cells. Now e mean say all the genetic materials, all the genetic content, everything wey dey inside the mother cell, we go divide am totally into the two daughter cells. Now the question na wetin be chromatid? Wetin chromatid be na chromosome wey don replicate, e don make another portion of itself, another version. If a cell wan divide into two now, all the chromosomes wey dey inside the cell, remember say the chromosomes dey been wrap themselves around the DNA. All these chromosomes, when they see say okay, the cell wan divide into two now, they go make another copy of themselves. So each chromosome, another chromosome go dey. So any two chromosomes wey dey like that, wey don come shorten together because they don dey ready to separate into two different cells, na wetin we dey call chromatids. So DNA dey, gene dey, chromosomes dey, and chromatids dey. Make sure say this thing no confuse you again. You understand am perfectly. Now after all these ones, the next thing we go talk about na the mitochondria. Some people dey call am the powerhouse of the cell. That one dey okay too, but the major thing wey you need to realize na say the mitochondria na the area for the cell where ATP dey generated. Wetin be ATP? ATP na wetin we dey call adenosine triphosphate. And wetin be the use of ATP? The use of ATP na say all the activities wey the cell dey carry out, if the cell dey do cell division, if the cell dey do digestion, any activity wey the cell dey carry out, the cell need energy. All the reactions wey dey happen for the cell need energy, and that energy na ATP be the energy wey the cells dey use. As the cells dey use up the ATP, they go dey use up their energy. So that ATP dey produced inside the mitochondria, and this mitochondria dey inside both plant cells and animal cells. These two cells dey get mitochondria. Mitochondria dey very important. For the structure of mitochondria, you get area wey we dey call inner matrix. That's inside the mitochondria, na the inner matrix, and na for there all the reactions dey take place for inside the mitochondria. Now the mitochondria get two membranes, two structures. So e get outer membrane and e get inner membrane, and all the reactions dey take place for inside the inner membrane. So the mitochondria dey very important. The next organelle we go talk about na vacuole. Wetin be vacuole? Now both plant and animal cells dey get vacuoles, but they get different types of vacuoles. Plant cells dey get wetin we dey call contractile vacuole, and that contractile vacuole e dey for a process wey we dey call osmoregulation. Wetin be osmoregulation? Now every organism, every plant need to dey regulate, dey control the amount of water and salt wey go dey inside the cell and wey e go push out. If water don too much for the cell need to push out some of that water so that the cell no go burst. And if water too small for inside the cell need to collect more water into the cell so that the cell go dey okay because of the many activities wey the cell dey do. So in order for that process to dey go well, to dey go on fine for inside the plant cell dey use wetin we dey call contractile vacuole. So that vacuole dey big, e dey contain water. If the cell need to reduce the water, and na plant cell dey get contractile vacuole, plant cell, if you need to reduce the water, you go go remove some of the water from inside the cell, and if you need to add more water, you go take more water into the cell with the contractile vacuole. That one dey for plant cells. Now for animal cells no dey get contractile vacuole. Wetin dey get na wetin we dey call food vacuole. And wetin that food vacuole dey for? Many animal cells dey engaged in wetin we dey call phagocytosis, meaning say they dey engulf different materials wey they dey feed on, whether na other animals or na materials like sugars or like glucose. So if they go engulf am, that thing wey they don engulf, wey they don swallow for inside their structure, they no go allow that thing make e dey run around for inside the cell. No, they will keep am for one area of the cell, form structure around that thing wey they carry enter their body. So that na wetin we dey call food vacuole. For inside that structure now, they go dey dump in wetin we dey call hydrolytic enzymes. Those enzymes go help to digest that structure or that organism wey dey inside that vacuole. So you need to understand say the plant dey get contractile vacuole, the animal dey get food vacuole, and this one dey very important. Next thing to discuss na endoplasmic reticulum. And wetin this one be? This one na system of membranes. If you look into any cell, whether na plant cell or na animal cell, they go get some membranes wey dey there. Now sometimes the membranes dey smooth, we dey call them smooth endoplasmic reticulum. Sometimes e dey rough and we go call am rough endoplasmic reticulum. The time wey e rough na say ribosomes bin attach to different parts of the cell, of the membrane of the endoplasmic reticulum. Na why we dey call am rough endoplasmic reticulum. So smooth dey, rough dey. And wetin be the function of these membranes wey we dey call endoplasmic reticulum? E dey plenty for inside cell. E get two major functions wey these membranes dey run. Now one of the functions na say they dey help for transport within the cell. If the cell wan transport material, enzyme, something from one end of the cell to another end of the cell, those things go dey transported through the endoplasmic reticulum. So that one na one of the functions. Another function wey the endoplasmic reticulum dey carry na say the endoplasmic reticulum dey form, e dey help as a site for all the different reactions wey the cell wan run. The cell need space to run all these different reactions, metabolic reactions, enzymatic reactions wey dey occur for inside the cell. The cell need space, need site to run all these things, and the endoplasmic reticulum dey provide that site for the cell. Say okay, come run all those reactions on top this area because space dey for this area. So na wetin endoplasmic reticulum be, smooth or rough. And those endoplasmic reticulum dey for inside both plants and animal cells. The other thing we go talk about too na Golgi bodies. Golgi bodies na small sac-like structures, and wetin they dey do? They dey help to package proteins. They dey important. If the cell don produce proteins, wan come package them, put them in a particular structure, na the Golgi bodies go help the cell to do that function. And those Golgi bodies dey for inside both plant cells and animal cells. This one dey very important. Then e get something wey we dey call chloroplast. Chloroplast dey, and chloroplast dey important because e dey carry pigment wey we dey call chlorophyll. Now chloroplast dey only inside plant cell. They no dey see am inside animal cell. E dey inside plant cell because e dey carry pigment wey we dey call chlorophyll. We go still discuss all of these ones later. Now this chlorophyll dey help plants to trap the energy wey dey inside sunlight, and if you trap that energy finish, you go use that energy take produce glucose and then every other material wey the plant dey produce. So chloroplast dey very important. E dey for inside plant cell. You no go see am for inside animal cell. That one dey important. Now we get another structure too wey dey call lysosomes. Lysosomes. Wetin lysosome be? Lysosome too na sac-like structure wey dey carry a lot of enzymes. Now these enzymes, we dey call them hydrolytic enzymes because these enzymes dey help digest a lot of things for inside the cell. The lysosomes dey present for both plant cells and animal cells. If anything may enter the body of these cells and they need to digest the thing, they need to break am down. Na those hydrolytic enzymes wey dey inside this lysosome, the lysosome go move towards that thing wey they wan digest, pour out some of those juices for that thing, and they go digest am. Another thing wey dey inside the lysosome too na respiratory enzymes. A lot of enzymes wey dey help for respiratory reaction wey dey occur for inside the mitochondria. A lot of those enzymes dey for inside lysosomes, and they dey keep them for there. Lysosomes dey for both plant cells and animal cells. The next structure na ribosomes. Wetin ribosomes dey do? Ribosomes na also sac-like structures. All these things wey I dey tell you now na organelles, sac-like structures wey dey inside both the animal cell and the plant cell. And wetin ribosomes dey do? Ribosomes dey function for plant synthesis. Wetin be the meaning of plant synthesis? Now we know say plant protein synthesis, protein synthesis. Now we know say protein na giant molecule. Now we dey call them macromolecule because e get small micromolecules wey go join together to make that macromolecule. You know the time wey I dey discuss DNA, I say small molecules dey wey we dey call nucleotides wey go join together to make the larger DNA. Na so protein dey too. Small molecules dey wey we dey call amino acids. If we link them together, we go use am form a giant macromolecule wey we dey call protein. So that process, the ribosomes dey help in that process, help to join these proteins together and join these amino acids together and then make protein. This one dey very important. Now cell wall. Ribosomes dey for both plant cell and animal cell. They dey for there. Sorry, I been get catarrh. Now for inside the cells too, e get wetin we dey call cell wall. This cell wall dey we dey find am for bacterial cell, but we dey find am also for plant cell. E no dey animal cell. Animal cell no get cell wall. Now many plant cells get cell wall, and the greatest constituent or the most important constituent wey dey this cell wall na cellulose. Na structure wey dey for inside plant. So cellulose dey this cell wall, and this cell wall get many different functions wey e dey do for the plant. E dey help the plant cell with strength and support. So e dey strong. If water full inside the plant cell, the cellulose cell wall go hold the water in well, so that the plant cell no go burst. So many other things wey e dey help give plant cell im own shape, give am im own size, give am structure, give am strength. These are the things wey the cell wall dey do for plant cell. But the animal cell no get cell wall. Now follow that by talking about cell membrane. Cell membrane dey for inside both plant cell and animal cell. Now cell membrane na little membrane wey dey around the cell, and we dey call am selective membrane. E dey select materials wey go dey go in and come out of the cell. E dey very important, and e dey present in both plant and animal cell, and e dey run that process of selective transport. E dey select wetin go come in and wetin go go out. So that one make it dey very important and very useful for the cell. The last thing wey we go talk about na the centrioles. Now centrioles dey be wetin we dey call granular structures, small structures, and dey dey present only in animal cells. Dey no dey dey for plant cells. And as dey dey for animal cells, wetin dey dey do for there? When the animal cell wan undergo cell division, the granules get these granular structures, that's these centrioles, dey get the function wey dey dey do. Dey dey help the cell out, help am run that process of reproduction, so that everything wey the cell wan do in terms of reproduction, e go go well. Thank you. I hope say you been listening to this lecture very well. Na 12 different organelles we don discuss, wey dey either for inside plant or animal cells, or e dey inside both, and we don discuss their function. Thank you as you been listening to this class, and have a wonderful day.